unmute on Zoom and assume that it is working. If it is not, give me a, okay. All right, hello, welcome. If you can't hear me, raise your hand, but I'm usually quite loud, so I don't think it'll be a problem. Yeah, so I'm Steph Weber. I'm gonna talk today about organelles without borders. So just a basic introduction, cells are the basic unit of life. So in biology, the like sort of the atom of biology is the cell. And so I and all of you are made up of cells. And so this is, here we go. So one cell is surrounded by a purple membrane, and then you can actually see a dark blue stained DNA here. And so we as humans are made up of cells, but cells are, the unit of life is universal, right? So plants, onions, are also made up of cells. They look very similar to human cells. And the worm, which is one of my favorite organisms, which you will become intimately familiar with in a moment, is also made up of cells. So we are going to sort of zoom in on cells for today. And so back in 1665, Robert Hooke looked through a microscope at a slice of cork. And so cork is just bark from plants. Where's my mouse? There we go, sorry. And he saw all of these little, what he thought were rooms. So he called them cella, because cell is Latin for small room. And he thought it looked like the rooms in a monastery. And so that's where the word cell comes from. And basically a cell is, the unit of life is a room. So I'm going to make the analogy to a Berenstain Bears book that I don't know about you, but I classically read, or my parents read to me when I was a kid. And so the cell has a problem. The cell needs to stay clean so that it can function properly. And so just like brother bear and sister bear, the cell needs to organize its stuff and keep its clothes in the dresser and its toys in the toy chest and everything in order, right? And so in the absence of organization, in a dirty room, the toys are strewn across the floor, which could create a tripping hazard. The dirty dishes are left on the table, and that could be a health hazard. And so Mama Bear is very upset and wants the children to organize their room. And so when everything is in its proper place, toys can be quickly found easily. There's no safety hazards. Everything is in its right place. And the cell has to do the same thing. So cells are organized into functional compartments, just like the dresser and the toy chest, etc. And those compartments are called organelles. And so it's literally the same thing as your body. It has different organs, and those are specialized, compartments that do a particular function. And so the cell has the same thing. So if I zoom in on a particular cell, there are all sorts of, in this case, membrane-bound compartments called organelles. Okay, so we're oriented. So now we're going to zoom in on a single cell. So this is a mammalian cell from a mouse. And what you can see is that there are all sorts of compartments, right? And so these compartments have specialized functions. So the nucleus maintains the genome, that's where the DNA is. The ER and the Golgi package and sort proteins, and the mitochondria make ATP, the energy of the cell. And so all of these specialized functions are made possible by membranes. So a membrane, it creates a physical barrier between the inside of an organelle and the outside of the organelle. And that allows each compartment to maintain a distinct chemical composition. And so the nucleus will have blue proteins or blue molecules that maintain the genome. The mitochondria will have blue or a different color molecule that makes ATP, etc. And in the absence of membranes or outside in the cytoplasm, outside of organelles, there's no barrier. And so molecules are just free to diffuse around and mix. And depending on the function of the molecules, that might be a bad idea. Okay? So for hundreds of years, maybe 150 years, membrane-bound organelles were considered the unit of intracellular organization. So cell is the unit of life. Organelles are the unit of intracellular organization. And that was because you can see membranes, you can see these barriers very clearly in electron microscopy. But in the last 10, 15 years, we now have better microscopes and we can use fluorescence microscopy and we can see all sorts of other compartments that don't show up on EM, so they don't have membranes around them, and yet they still are concentrated local regions of space that have particular molecules that are still in high concentration relative to the outside. So, so there are a variety of different flavors, if you will, of these organelles. There's germ granules, so there's some in embryos, there's some in skin cells, in neurons, they're in the cytoplasm, they're in the nucleus, they're everywhere. And yeah, and so this was like crazy, right? Because in the cytoplasm, there's no organization, everything can diffuse around, there's no barriers. So how do these things exist? So that was a huge question. And going from electron microscopy, where you look at like fixed cells, like single snapshots in time, with fluorescence light microscopy, we can look at movies. And so this is why I'm a cell biologist, for the movies. So in this particular, this is a worm embryo. And what we've labeled is a protein that localizes to germ granules. So it's eventually important in defining the fate of a particular cell. So this defines which cells differentiate into germ cells, egg and sperm, and all cells that don't inherit this protein become somatic cells. And so what you can see is that there are concentrated spherical looking things that I will call droplets. They initially start uniformly in the cell. They sort of dissolve or disappear from the anterior. They condense or stabilize in the posterior. And then this posterior cell becomes the germline. And so, so these membraneless organelles, they're dynamic in response to development. And they're also dynamic in response to stress or external environmental conditions. So here you have a human cell that is expressing a protein that is important for stress response. And so when they add stress, this protein is initially uniformly distributed in the cytoplasm, then it condenses into these, sorry, I'm asynced with the movie. There, they add the stress, the protein concentrates into these droplets. You can see some of these droplets forming spherical things that then fuse and form larger spheres. And it's reversible. So when the stress is washed out, now, then the droplets disperse, okay? So, and this is super cool because if you wanted to make a membrane-bound organelle and get rid of a membrane-bound organelle, that would take a lot of energy and probably a lot more time than we have here. And so membraneless organelles allow cells to respond very quickly to developmental or environmental cues. But we still have no idea how this works, right? So we have a system that has no boundary, and yet it still is able to concentrate specific molecules. So that is the question that was posed a little over 10 years ago that ushered in a new phase in cell biology, and is a pun that is terrible, and it will become more obvious in a second. So, okay, so about 10 years ago, people took movies of these membraneless organelles and noticed that they looked like liquids. So this is, these are nucleoli, which is a particular type of membraneless organelle. And if you have two of them and they come into contact, then they round up. Just like if you had like two raindrops on your car windshield, right? And then over here, what we're gonna look at is those same germ granules, but instead of in the embryo, it's in the mom before the embryo is formed. And in the center, this black circle is the nucleus. And these germ granules are like adhered to the surface of the nucleus. And then what's gonna happen when I play this movie is someone is pushing down on the glass cover slip. And so they're applying a stress, a force, and that's gonna cause the germ granules to, so it's moving, but so they drip and they like flow off of the surface of the nuclei. So I saw these movies when I was a graduate student, and I was like, oh my gosh, I've never seen anything like this in a living cell, right? and it was amazing. And so it led people to start thinking of these membraneless organelles as liquid phases. And so if we think about liquids, here are two examples that you are probably familiar with. So if you have a mixture of two liquids, they can either mix, like alcohol and water in beer, homogeneous, or they can separate. So oil and vinegar in salad dressing or in bread dip, they separate into a vinegar, high concentration of vinegar, high concentration of oil, and there's no physical barrier between them, but they still separate. And so we can have liquid matter that is both homogeneous, like when there was no stress, and that can separate, like when there was stress. And so this was sort of a conceptual model to start to think about what these membraneless organelles are doing in cells. Okay, so the other thing, I'm going to get a little bit technical here. Hopefully, I won't lose you. If you have a question like in the middle, I'm happy to answer it and make it interactive, by the way, if you want. Okay, so membraneless organelles, they are made up of proteins, which are just molecules that do a lot of things in the cell. And the proteins that make up membraneless organelles have specific features called multivalency and disorder. And I'll go through these. So multivalent protein, many, and a valency is the number of domains that can bind to a specific thing. So in this particular case, in this cartoon, there are 3 domains that are the same shape. So the valency of this molecule would be 3. The valency for this shape would be 1. And so it's a repeated modular domains. And the more valency you have, the more ability you have to bind to many things. Also, proteins can have what are called intrinsically disordered regions. So most proteins fold into a stable 3-dimensional structure. Proteins that have what are called low complexity sequences, this just refers to the sequence of amino acids or the sequence of building blocks in the protein being not highly repetitive, or being highly repetitive, not. So like GYG, SYS, lots of repeated letters. And what that does is it causes them to form extended flexible conformations. So here's just a little movie. In this protein, you have an ordered domain here. So this would kind of be like a valency of 1. And then you have these IDRs that are these like floppy tails that hang off. And these floppy tails can interact, and the binding and the folded domains can interact. And so if you have many high valency binding domains, you can form many interactions with many different partners. And if you're flexible, then you can form many different partners with many different molecules. And the point of that is if you think about, like, if I'm a human and I try to hold on to someone else's hand, I can only hold on to two hands max, right? And so I've got 2 people, but I can't form a network out into a larger area. But if I had a 3rd hand, then we could start to build something larger. Yeah. Yeah. No, it's a great question. Yeah, so the other proteins and sometimes nucleic acids, so DNA, RNA, but other molecules that themselves may be multivalent and disordered. No, much smaller. So there are millions of these inside an egg and a sperm. Yeah. Okay, so what we observe is if you take these proteins, molecules, that normally in the cell are found inside these membraneless organelles, and we purify them and have them in a test tube, then they form these big, beautiful spherical droplets, or they form these kind of like jello, solid hydrogel things. And so basically what you can think about is if you just had a valency of 1, then it would just be a solution, a liquid everything would be light green and nothing would form these condensed phases. But because they have multivalency, this one purple molecule can interact with 3 blue molecules, and that allows them to form this large network that holds this condensed liquid phase together. And if the intermolecular interactions between the different molecules or between the different disordered regions are really strong, then you'll form a hydrogel because they won't unbind. But if they're weak, then they can unbind. Like I can hold your hand and then let go, and then hold someone else's hand, and so the molecules can move around. And that's what gives liquid phases their fluidity. Okay, so all of that is prefaced to state our hypothesis. So when I started in this field, we hypothesized, based on the evidence that I just discussed with you, that membraneless organelles assemble through an intracellular phase separation. And the idea, the concept is, okay, we have soluble molecules, and if they're, univalent or bivalent, they won't have enough hands to hold each other together. But if we increase the valency or the low complexity, then now there's enough weak interactions to pull and hold the molecules together, even if they're transiently binding and unbinding. They'll still come together in this high concentration region. And then if we continue increasing these interactions, then suddenly they become too strong and they can't let go and they can't rearrange. And so they form these solid, in this case, fibers. And we hypothesize that this might be involved in sort of aggregation, which we'll get to in a second. But there's a lot of neurodegenerative diseases where proteins aggregate. Yes, exactly. Great. So we put forward this hypothesis that molecules that are soluble in the cytoplasm behave like gases. I'm not suggesting that there are air bubbles in your cytoplasm, but that the molecules are relatively low concentration and free to move around anywhere. Or they can exist in a liquid state, which are like those droplets of the movies I showed you earlier. Or they can irreversibly bind really, really tightly, and then the molecules will not be fluid, and they will just stay there. And depending on what they do, that could be bad. Okay, so that's hypothesis. And people have shown, indeed, if you purify proteins, they can span this spectrum of material properties. You can get liquid droplets, you can get amorphous fibers, and this is still droplets, but you would get a uniform gray box if they were soluble. Okay, I have one physics slide, and then we'll get into the data. So, what is phase separation? It is a thermodynamic process by which a homogeneous solution spontaneously demixes to form 2 or more coexisting phases. So if you think about it like salad dressing, you shake it up really vigorously, then it is homogeneous, and then it spontaneously demixes into an oil phase and a vinegar phase, right? And those coexist; those are stable at equilibrium. And so you can predict whether a system will phase separate or stay mixed, right? The beer stays mixed, based on its free energy of mixing. And so the free energy for any system is based on the sum of the entropy, which is the measure of the disorder or the randomness or the number of possible conformations that a system can occupy, and the energy, which is a measure of how strongly either attractive or repelling molecules interact. And so entropy always wants to mix, because the maximum disorder is when there's a homogeneous mixture. If I separate these things, I'm imposing order on the system. And so that's always unfavorable entropically. But if the energetic gain of separation can exceed the entropic cost of creating this order, then the system will still phase separate. So we have to think about the number and the relative strength of the interactions. So if the interactions of two unlike molecules is favorable, then it will always stay mixed because the entropy and the energy are promoting mixture. If the interactions between like molecules are stronger and really stronger than the unlike molecules, then if that exceeds enough tropic cost, then it will phase separate. Yep. Yeah, so this is just, I mean, this could be alcohol and water. This is not really biology. This slide right here is physics. And the biological context is everything. And I'll give you some more specific examples in a moment. So it happens during development. It happens during growth. It happens during aging and death. Phase separation has been observed in basically all biological contexts. Sure. Yeah. And I mean, one of the really exciting things about this is that it's so versatile. Just by creating this organization, you can organize many different types of molecules to do many different things. And so it has been observed in many different biological contexts. The running joke is, oh, it phase separates. Everything phase separates, so it's meaningless, but it can do a lot of things. Okay, so whether it mixes or phase separates depends on the relative number of interactions and the differences in strength. And so we can plot this on what's called a phase diagram, where on the x-axis we have concentration. So over here we have a lot of blue molecules. Over here we have a lot of green molecules. So if we have more green molecules, then we will have more of these, in this case, favorable interactions. And so it will phase separate in this intermediate regime. I'll go through a more specific example in a moment. Yes? Yeah. Yeah. Right. So oil is hydrophobic. So it's sorry. Oil prefers to interact with oil than it does with water. And so what that means is that green, let's say oil is green and water is blue. So these interactions will be stronger and more favorable than this interaction. And so that will promote phase separation. But if in the case of oil and, in the case of alcohol and water, this is actually more favorable. Well, it's not actually more favorable, it's about equal favorable. And so the entropy wins, and so it mixes. Yes. Yeah, great. Okay, so this is what you would see in a physics class. And it's really cool because we can write down the equations and make this prediction. But the real question that I had was, does phase separation, does all of this beautiful, thermodynamics actually occur in a real living cell, right? And so what we use is this membraneless organelle called the nucleolus. So this is in a human cell. Here are these green blobs in the nucleus. And so this organelle functions to make machines in the cell that make more protein. So it's required for growth, for cell growth. And okay, so we won't worry about the DNA. So we chose C. elegans, this worm, to study this process, mostly because the embryos are small and transparent, and we can watch it in movies. And so here we have the red is the cell membrane, and in green I have a protein that is in the nucleoplasm or in the nucleus, this membrane-bound organelle, and then it concentrates into two liquid droplets that can sometimes fuse, and so here you get one big one. Okay, so now I'm going to go through the phase diagram in detail and then show you how it compares to experimental data. So, so this is a phase diagram where the y-axis is temperature and the concentration is on the x-axis. And actually, sorry, let me go back. So temperature here is it. Well, temperature is in our equation for free energy. And so if I increase the temperature, I'm going to favor entropy. And if I decrease temperature, I'm going to favor the energetic component. So up here, I'm going to favor entropy and favor order and mixing. And down here, I'm going to favor energy and phase separation if the interactions dictate that. Okay, and so let's take a very simple system of two liquids, red and blue. We start out with a red, with all red molecules. We add a little bit of blue. Nothing particularly exciting happens. It's still mixed. But if we cross this magic concentration, this phase boundary, then suddenly we'll start to see droplets of blue in the sea of red. And if we keep going, now we're going to add more and more blue. They're still stay phase separated, and so the droplets will just get larger in volume. And then eventually we'll have a 50-50 mixture. And then if we keep going the other way, now we'll have red droplets in a blue background, and they'll get smaller as we increase the concentration of blue, effectively decreasing the concentration of red. And eventually we will get to a mostly blue homogeneous mixture. If we increase the temperature, then even we're going to favor the entropy over the energy. So even if the red and blue don't particularly like to be together, if we increase the temperature enough, eventually they will mix at all concentrations because we're favoring the entropy so much. And so in this phase diagram, we have two regions, one outside of this phase boundary that is a single phase and it's always mixed regardless of the concentration. Inside this phase boundary, we have a separated system where there are two phases of mostly red and mostly blue. Okay? And so from this phase diagram, we can pull out two features that then I can test experimentally. So first, the saturation concentration, the concentration at which you first cross this phase boundary, sets a threshold. So below that saturation concentration, you don't see any droplets. Everything is homogeneous. Above that saturation threshold, you get droplets. So it's a threshold. And then the second thing is that above the saturation concentration, the size of the condensed droplets should increase with concentration. So we increase the concentration of blue, they get bigger. Here, experimentally, I can only explore a very narrow range, so we won't actually get to the other side. Okay, everyone comfortable with the phase diagram? Oh yes, Ruby. That is a fantastic question. So it's more a technical challenge, and so we're microscopy, and so there may well be micro droplets below the resolution that we can actually detect, but I'll show. Hopefully, it'll be more obvious when I show you images. But we basically say that there's a droplet when the fluorescence, when the concentration, brightness of molecules is high enough above our limit of detection, the background. And that is not, that is a technical limitation, not a theoretical limitation. There's almost certainly, we are almost certainly underestimating the saturation, or sorry, overestimating saturation concentration. Excellent question. Okay.
Energy, the way force of some kind is generated through this process of mixing to
Yeah, so first of all, everything that I've been talking about, I'm not talking about mixing of organisms, I'm talking about mixing of molecules inside individual cells. So we're zoomed in. And so what is mixing are collections of molecules of, in this particular case, red molecules and blue molecules. So it's different, two different sets of molecules inside a cell. Or synthesizing other molecules. So I glossed over this, but the nucleolus, the one that I'm going to show you, this makes, its function is to synthesize ribosomes. Ribosomes are large molecular complexes in cells that make proteins. No. So let's say we want to move this big desk. I can't move it alone, but if I got 2 or 3 of other of you and you're got big muscle, together we could do something that an individual couldn't. And so all this phase separation is doing, well, one thing that this phase separation is doing is it is bringing multiple molecules together, and that at that high concentration, they can do something that they can't do at a low concentration.
That joining of these molecules generates heat or energy.
Or a conformational change, and then it's active. Like an enzyme can't act on its own, but when it's together in all of these other molecules, it becomes active because it changes shape. Yeah, I mean, in my silly analogy, it's pushing. In this case, it is transcription. So it is a molecule adding nucleotides to a longer polymer to create a new molecule. I feel like I'm getting further away from the question. Sorry? Yeah, so it's like maybe another analogy. Like if we didn't have lanes on the highway, there would be chaos and accidents, but if all of the cars are in the right lane going in one direction, then they can drive more efficiently than if there were no lanes. And so you can think about the nucleus as a lane, or as like it's organizing the space inside the cell so that the molecules in that particular space can do their function more efficiently. The functions can be very varied, and I'll show you some examples at the end of the specific functions. Yes.
But they have a very big difference, kind of like separated, like the top, so they don't naturally congregate together. But then there's natural separation that happens.
Sure. Yeah, so that's like the affinity, the energy of the arts people will be stronger than the energy of an arts person with a computer scientist, and they'll probably have a more productive conversation with other people who care about what they're talking about than with the nerds talking about computer science where they're just not interested. Does that help? Sure.
And I think the question is, if the 2 different disciplines, something important happens, something crucial happens to what you're trying to tell us, because in your analogy, driving somewhere, and these, what is happening here?
I mean, so yeah, yes, in this particular case, new things are being created. It could also be, so if we go back to the arts and CS, If you had all of them together in a big mixture, then you wouldn't have a productive conversation because the computer scientist would be trying to talk about X and the artist would be trying to talk about Y. And so if they separate, then they can each have a more efficient conversation. And so this isn't a conversation, this happens to be synthesis of a new molecule. And so not only are the enzymes that make the new molecule concentrated in this droplet, the subunits, the molecular things that they need to make the new thing are also concentrated in these droplets. I'm just not showing them. All right. It's a great question. I'm glad you're thinking. It's good. Okay, so I wanted to test this hypothesis, or I wanted to see if there was a saturation concentration, and which created a threshold, and if the size of the droplets depended on the concentration. Those are the two things I'm trying to test experimentally. Okay, so a convenient thing that I'm not going to go into the details about how I do it, but I can change the I can manipulate the concentration of all of the proteins inside the nucleus by changing the size of the embryo. So small embryos have a high concentration. So this is the concentration of my favorite protein in the nucleus, and this is the embryo volume. And so I can genetically manipulate the size of the embryo to create embryos that have high concentration of these proteins or low concentration of these proteins. And if I look at an embryo, a control embryo, it has two droplets of some size. And then if I look at the mutants, I have a big embryo that has a really low concentration and really tiny nucleoli. And I have a small embryo that has a really high concentration and they have giant nucleoli. So Ruby, to get back to your question, if these were just a little bit smaller, I would not be able to detect them. So it's the difference in concentration or the brightness. Okay, so if we want to measure this quantitatively, we're physicists, then we can look at the integrated intensity, the brightness of this image. And so this is just a three-dimensional plot of this bright white spot is this high relative to the background. And so in each cell, this is 1, 2, 3, 4, 5, 6, 7, 8 cell stage. So in each cell, there are 2 nucleoli. And so this one happens to be a little bit smaller than this one, but I can sum up the intensity inside this object and get the intensity of one nucleolus, the intensity of the second nucleolus, and then I can calculate the maximum intensity of nucleoli per cell. And I pull them together just because there can be some variation between the two. That it actually turns out to be an interesting question, but for today, we're just going to sum them together. Okay, so now here I have, I was able to change the concentration, right? So I've got my concentration on the x-axis. On the y-axis, I have the nucleolar intensity, which is just a proxy for size. And so at really low concentrations, I have small nucleoli. At really high concentrations, I have large nucleoli. And so I do indeed get a proportional increase in size as a function of concentration. So that's super cool. That's my second property, right? And so we can compare this quantitatively to a model for phase separation, which relates the size of the organelle, so this is the intensity of the organelle, to the difference in concentration between the concentration in the nucleoplasm and the saturation concentration, times whatever the size of the box, in this particular case, is the volume of the nucleus. And so what that turns out, so we can measure, the alpha is just the intensity per molecule, not important. So we can measure each of these things independently and then calculate and predict what the organelle size should be. And that is what I get. And this is not a fit, this is a prediction. This is like the best prediction that you can ever ask for in biology. Like it actually goes through the points. It's amazingly accurate. Biology is messy, it's not physics. Okay, so here, because even at the lowest concentration, we're above the saturation concentration. So they're small, but they're still there. And so ideally, to test my first property, I would decrease the concentration a little bit more, and then the prediction would be is I would see no nucleoli below the saturation concentration. I couldn't do that. If I'm trying to manipulate these embryos genetically, and I couldn't go any further to the left. Very disappointing. But like, yeah, yes, I could, yeah, and I will show you I can in a roundabout way. So, okay, so hold that thought. So just to graphically show you, hopefully to orient you. So what we're plotting in this model is the concentration in the nucleoplasm is something here, and then the saturation concentration is here. And so this term is what is the degree of supersaturation. So it's above the saturation concentration, and the further to the right I go, the bigger, the more above it we are, and so the larger the droplet size will be. Okay, so now getting back to the question, how do we go further? How do we test the threshold hypothesis? And we couldn't at this particular cell stage, but the embryo develops and goes through many different cell stages. And so everything that I've shown you so far was at the 8 cell stage. At the 4 cell stage, for reasons that I have no idea, but was experimentally useful, nucleoli don't assemble. So in the wild type, normal, unmanipulated embryo, there's no nucleoli at the 4 cell stage. There's some really tiny, small ones that I may or may not be able to detect depending in this cell type. But in these 2 cells, not important what they are, but in these 2 cells, I never see nucleoli at the 4 cell stage. So this is great, right? Because now I can do the same thing, and if I decrease the concentration even more, then okay, I still don't get nucleoli, fine. But if I increase the concentration, then I can nucleate nucleoli in a stage where they don't normally assemble. And so I have a saturation concentration above which nucleoli assemble and increase in size, and below which they don't assemble at all. And so what happens is, and I don't know why this is the case, this is just biology, but the saturation concentration at the 4 cell stage happens to be higher than it is at the 8 cell stage. I don't know why, but it's very convenient. And so you can see that actually the this middle condition, this red condition, is roughly at the concentration, the saturation concentration. And so about 50% of these embryos have small nucleoli, and about 50% of them don't have any. So that's like right. So that's basically my limit of detection. Okay, so now we can go from my cartoon phase diagram to an actual phase diagram of a living cell, or of a nucleus in a living cell. Yes. Yeah, so my goal in doing this was curiosity. I thought this was super cool and fun, and I had no application in mind. Yeah, I wanted a PhD, or actually this was my postdoc. Anyway, yeah, so, but there are many applications of phase separation that are now coming apparent with more and more examples and more biological context. And I may jump to those at the end. Let me go through this one slide, and then I'll show you the biological context and the. Okay, so on the x-axis we have nuclear concentration. On the y-axis we have a completely made up. I don't know what it is. I'm hand waving. Developmentally regulated parameter, where somehow the 4 cell stage the molecules are less sticky than they are at the 8 cell stage. I don't know. That's made up. But what I can do is. I can make this semi-quantitative by using the experimental data for the x-axis. And so at the 8 cell stage, the saturation concentration is something, and all of the cells are above that, and they all, so this gray line here is equivalent to this gray line here. So as if it's coming out at the at you. And at the 4 cell stage, there is a different interaction parameter, and so it moves up, and that changes the saturation concentration, but the concentrations of the molecule of the protein in the cell doesn't change. But now the saturation concentration is in the middle. So these cells don't assemble, these cells do. And these, the further into this phase separated region, the larger they are. Okay, I'm gonna jump wildly to the end because I wanna give you some context. Sorry. Okay, so intracellular phase separation is ubiquitous. What I didn't show you is, so this is the example of the nucleoli in worms. We've also, my lab has found phase separation in bacteria, which I think is super exciting. And there are examples in humans, there are examples in yeast, there are examples in plants, they're everywhere. And so then the question is, what do they do, et cetera. But so they are so ubiquitous and they are really becoming, it's not just membranes that organize the cell, like we are rewriting textbooks. Membraneless organelles, phase separation is a fundamental and essential process that organizes cells. So that's the punchline if you take anything away from tonight. Now, what do they actually do? Fantastic question. The joke is we don't know, but I'll try to give you a couple of examples that we kind of sort of maybe think they do. So membraneless organelles have been suggested to have many different functions. The most obvious is what's called a reaction crucible. So this is what I was talking about before. If you increase the concentration of molecules, if they perform a chemical reaction, that reaction will go faster if you increase the concentration. That's just like standard chemistry. If you increase concentration, the rate, the total rate goes, sorry, the rate constant is the same, but the total rate of the reaction increases. So an example here is if you have this protein that phase separates NWAS, NWAS happens to bind to a protein called actin, and when it's actin alone, it takes a really long time for these polymers to form, but in the presence of NWAS, then they form much more quickly. So it can accelerate things. It can actually do the opposite. So if I pull blue away from red, but actually the productive thing is red and blue interacting, then I can inhibit a reaction. I can stop something from happening. And so there's a sequestration function where these stress granules, when I showed you before in response to stress, cells want to stop making new things because they might make a mistake in those new things because it might be damaged. And so they sequester these proteins away from their target so they can't act on a damaged substrate, for example. Another one that is really common in the nucleus is as organizational hubs. And so this protein called mediator is important for transcription, for turning on genes, activating genes in the nucleus. And the cell has, many genes encoded in DNA at different regions of this long polymer. And what these condensates, or what these this phase separation can do is it can bring different genes on different chromosomes together, and so they can activate gene expression together. And so it can coordinate gene expression on, for example, 2 different pieces of DNA. There's your context. And then why would we care? The application is this process, phase separation in general, in many different contexts, has been shown to, or has been implicated in disease. So in breast cancer, the size of the nucleolus, see, it matters, can predict the prognosis of patients. So malignant tumors have enlarged nucleoli, whereas benign tumors or normal non-cancerous tissue has small nucleoli. And so either there's an increase in concentration or an increase in activity. We don't exactly know why, but this is diagnostic of, or it's used by pathologists to diagnose cancer patients. About 10 years ago, there was this ice bucket challenge, if you guys remember, to raise money for ALS. So the protein that I showed you before that formed that jello-like hydrogel, it forms liquid droplets in the cytoplasm in these in patients with ALS, and there are mutations in the low complexity domain that cause it to be more sticky, and then these aggregates are correlated with motor neuron degeneration. And then the potential exciting thing is that we could actually use phase separation for treatment. So this is very new. This is not in humans, anywhere close to humans, but some drugs concentrate within the condensates. So cisplatin is a chemotherapeutic, and it concentrates inside these MED1 transcriptional droplets, but not in nucleoli. And so it might be able to act on these, specifically on these droplets. And so the idea is that we could design our drugs to preferentially concentrate in particular droplets that we know about, and that could either target the drug to only act here or only act on cells that form these droplets, or there are other kinds of drugs that actually dissolve the condensates or the droplets. This process, phase separation, has also been used to design synthetic organelles. So we can engineer these floppy or modular protein modules onto other proteins, and we can form, synthetic non-native droplets. And in this case, these droplets do different biochemistry, and so you can actually make a cell make proteins that it wouldn't be able to make in the cytoplasm. And then it has also been implicated in the origin of life. So some of you may be familiar with the concept of an RNA world. So RNA probably predated proteins, and RNA can phase separate with really simple peptides that would have been on primitive Earth, prebiotic Earth, and that forms these droplets. And there is some theoretical evidence that suggests that droplets can grow and divide on their own. So I will leave you with a quote from an 1899 paper, way before all of this, that said that all elements of the protoplasm, which is the cytoplasm outside of the membrane-bound organelle, are liquid. And so E.V. Wilson is a famous biologist, and he looked at this image of a sea urchin egg, and he saw all of these spherical little droplets. And he thought, oh, that looks like, droplets of various things. Probably turns out that it was like the oil from the yolk, and not necessarily what I've been talking about, but it's a nice full circle. So intracellular phase separation is a new mechanism for generating functional compartments in living cells. It allows cells to respond rapidly to internal or external cues. It could serve as a target for disease therapy or as a tool in therapy, and it could be harnessed for synthetic biology. And so with that, I will thank members of my lab, and if you have any more questions, and sorry if I ran long.
So at the very end, when you said this was a new mechanism, you are it's not new.
I absolutely understood mechanism. Yes. That. No, I'm sorry, I couldn't. No worries. We can chat after if you want. Yes.
Concentration, there's also the level of energy. Is the level of energy involved in cells typically in either encouraging phase separation or unphasing the proteins?
That's a fantastic question. So the physics of the thermodynamics of phase separation assumes equilibrium. And the cell is not in equilibrium. Equilibrium is death in biology. So actually a lot, and quite frankly, many of the predictions from equilibrium, it is surprising that works at all, and that it can describe what we see because it is in a non-equilibrium system. So the energy of what I showed there was like basically like the energy of interaction. So how sticky or how attractive or repulsive. Yeah, that is all totally within the sort of on the order of thermal energy. So that's legit. What on top of that, what biology has is active enzymes that like break bonds in ATP that gives you above thermal energy. And that brings you to an energy scale that's a little bit higher, which actually might be how biology controls and uses phase separation. And so to go from this homogeneous mixture to the condensate or to the droplets, you have to change something. Otherwise, at equilibrium, it wouldn't change. You wouldn't be able to respond. And so it's almost certainly active non-equilibrium chemistry that happens to transition from the homogeneous to the condensed state or back.
Yeah, that's what I was going to ask. You talked a lot about the condensed state, but obviously in some cases you want to go back.
Sure.
Otherwise, it's a one-way street.
Yes. No. Exactly. And it's almost certainly an active process to go to whichever one is not the natural. Yeah.
We're going to take a question from online, and then we'll go.
Right. Yeah. So one question that we got from online is, what is the importance of membraneless organelles in or for viral infection?
Yes. So I didn't talk about it, but many proteins that viruses encode phase separate. And so one of the new things that came out in the last year or two is that the spike protein in COVID can phase separate, and that is thought to help the virus replicate and then form new viral particles. And so if you mutate this protein so that it can't phase separate, then it doesn't in cells. This was clearly not released into the wild. Then it can't propagate in cells. So it's again, that could be a target for therapeutics eventually. On a longer time scale than vaccines.
All right, thanks so much. And yeah, we can go to your question.
So you explained how phase separation creates organelles in cells. Can it also explain how in developing organisms, like organs are actually formed, like when the fetus is the embryo is developing?
Yeah, that's a great question. So yes and no. So yes, the physics, the description of the physics of like things coming together and separating from unlike things, people have used the same equations to describe cells, like cell types coming together and separating from other cells. And it actually works pretty well. But the interactions, they're not molecular scale interactions. They're like protein-protein interactions on the cell surface that are binding to each other. And so they're much higher, much stronger energy, but also much larger things that are moving around and sorting. So yes, the concept and the equations work out, but the molecular details totally different.
Getting down to very, very basic. In each cell of anybody, of any organism, chemical processes are going on. Wouldn't there be a huge number of different molecules, a very, very large number?
Yes. Yes, I totally simplified it as in there are two. In reality, there are millions, and there are, all of these membraneless organelles have dozens to hundreds of different molecules that concentrate in them and not elsewhere. But it's already confusing with two, and so. But yes, it is absolutely more complex than the picture I described, for sure.
You have a question?
Yes. In your experiments, do you or when you be able to like go back in like in the you created the nucleoli with different concentrations, but could you or you try to do it like the other way, like because it's reversible, right?
Yeah. So with changing the concentration, we weren't able to like dilute them. But we have a different mutant that I didn't talk about, that where nuclei don't assemble, even at the 8-cell stage when they always assemble. And so basically what we're doing is by doing this mutation, we're weakening the energy of interactions between not just the green protein that I looked at, but many others. And I was convinced that these embryos would have some defect or wouldn't be able to hatch or wouldn't be, they're fine. So I don't know what's going on, but that was a really disappointing result. But yeah. So we're sort of using this as a test tube to understand what the molecular interactions are that bring these things together. And hopefully eventually we'll be able to come up with a manipulation that has a biological effect. But the truth is we're not there yet.
Yeah, sorry, I have a question about the intrinsically disordered regions and the ordered domains.
Yeah.
Like they're associated with phase separations, but are we talking about like different types of proteins with like different levels of IDRs? Or are we talking about like the same protein, but they change their level of IDRs or like maybe change of conformation like kindred? Like
yeah, mostly the former, but a lot of the disease mutations do the latter. So in general, if you look at like the whole proteome of like 20,000 proteins in the human proteome, the proteins that naturally have multivalent or disordered regions tend to be found in membraneless organelles. But in various disease states, mutations often occur in the intrinsically disordered region, or duplication may increase the valency, and then that causes aberrant phase separation. So both occur, but naturally it's more the former. Yeah, good question.
Okay, sorry. So I think we have maybe one good question from the online audience to end on. So this will be our last question, but it is, do you believe this process is so universal and efficient that it could also be present in life on other planets? Life on other planets.
Wow, that is super cool. To be honest, I have not thought about that. I mean, but basically, yeah, it's chemistry and physics, right? Like there's nothing unique about Earth or that. And it's been implicated in the origin of life here, so why not life elsewhere? But I mean, I have no idea. There's certainly no experimental evidence supporting that hypothesis, but it's a great idea to think about.
All right, thank you so much. So yeah, that was the talk. Thank you again, everybody, for coming out.